All right, everyone, welcome to my Chapter 7 presentation. I'm Patricia Stova, and this week we're going to be talking about the skeletal system. Now, the skeletal system has main 5 different functions. It moves the skeleton, maintains body posture and position, supports soft tissue, guards entrances and exits, and maintains body temperature. And just to get a little more in depth about each of these, so when we talk about moving the skeleton, we talk about when like our skeletal muscles contract, they pull on our tendons and they move the bones. This contraction, it may produce like simple motion, like extending your arm, or like highly coordinated movement, like swimming or skiing or typing. We talk about maintaining posture and body position. Continuous muscle contractions maintain body posture. Without this constant action, you could not sit up. You could not sit upright without collapsing or stand without toppling over. And when we talk about supporting soft tissue, we talk about how like the abdominal wall and the floor of the pelvic cavity consist of layers of skeletal muscle. These muscles support the weight of our visceral organs and shield our internal tissue from injury. And second to last, we talk about guarding the entrances and exits of our bodies. So skeletal muscles encircle openings, the digestive and urinary tracts, and these muscles give us voluntary control over swallowing, defecating, and urinating. And last but not least, maintaining body temperature, and that is something that we talk about in almost every single presentation because it is so important to the body. Muscle contractions use energy, and whenever this energy is used in the body, some of it is converted into heat. And the heat from the working muscles keeps body temperature and the range required for normal functioning. So all very important. And when we talk about skeletal muscles, skeletal system, we talk about 3 different types of muscle tissue. We talk about skeletal muscle tissue, cardiac muscle tissue, and smooth muscle tissue. And when it comes to skeletal muscle tissue, the cells are more like long, cylindrical, striated, and multinucleate, which means that there are multiple nucleuses. It's combined with the connective tissues and nervous tissue in the skeletal muscles. And this skeletal muscle tissue, it moves and stabilizes the position of the skeleton. It's going to guard entrances and exits, like we were saying. And yes, and if we look right here, oh, I can't draw. No. Oh, well. But if we look right here, it's more of cylindrical. You can really see the striations. You can see the multiple nuclei, and you can see the muscle fibers. But if we go to cardiac muscle tissue, we see how they're narrower. So we see how there's nuclei, and we see that there are like the muscle cells, but we also see the striations and the intercalated discs, which are kind of like little connecting ports right here. So the cardiac muscle tissue, the cells are shorter, branched, and striated, and they're usually with a single nucleus. The cells are interconnected by intercalated discs, and the location is going to be in the heart. It's going to circulate blood and maintain blood pressure, and so that's why the cardiac muscle tissue looks like this. It's because, heart, blood vessels, you get the idea. And the third type is a smooth muscle tissue. And these are going to be cells that are short, they're spindle-shaped, and they're non-striated, and they're only going to have a single central nucleus. And these are like found in the walls of the blood vessels and in the digestive, respiratory, urinary, and reproductive organs. It's going to move food, urine, reproductive tract secretions. It's going to control the diameter of respiratory passageways, and it's going to regulate diameter of the blood vessels. So we talked about how the cardiac muscle tissue was for the blood and for the heart. The smooth muscle tissue is going to be in the blood vessels in the rest of the body. So we see over here, they're shaped a lot like the cardiac muscle tissue, but we don't see the intercalated discs, and we see the smooth muscle cells because we don't see that we don't see the striations, the lines. All right, and so just a few keywords to keep going. We have the muscle fascicles, which are going to be like a bunch of muscle fibers. And muscle fibers are actually cells in skeletal muscle tissues. It's a singular muscle fiber. Each cell is a singular muscle fiber. And then last but not least are tendons. And tendons are bands of collagen fibers that attach skeletal muscles to bones. And then they're going to connect different skeletal muscles. The tendon fibers are interwoven into the periosteum of the bone, providing a firm attachment. So if we look at the diagram right here, we see that we have the bony attachment, we have the tendon, this little white part, and the muscle, and the tendon is connecting the two. All right. So right here, we can see that we have skeletal muscle. And from superficial to deep, different types of connective tissues are going to be the epimysium, the periosteum, and the endomencium. And we see them in this first diagram right here. The epimecium is like this very outer layer. The perimeum is going to be like this inner thing, this inner pink part that's kind of in between all of the bundles. And the little bundles, we see that's where the endomencium is. All right. And so there's different, we have different, here we go. Okay, so here is the muscle fiber. And we see that there's, different structures, different words in here. So the plasma membrane, we're going to call that the sarcolemma. It's going to be all the, yes, so the plasma membrane is going to be called the sarcolemma. We see right here. Yes, this little like red line surrounding the inner of the muscle fiber. And then the muscle fiber that surrounds the cytoplasm is going to be called the sarcoplasm. So, sarcoplasm, this white thing that's kind of surrounding all of the myofibril. And the openings that are scattered across the surface of the sarcolemma are going to lead into a network of narrow tubules called the transverse tubules or the T-tubules. All right, inside of each muscle fiber, branches of the T-tubules, they're going to encircle cylinder shape structures called the myofibril, and we talked about that earlier because the sarcoplasm is going to be what's surrounding all of them. All right. The sarcoplasm reticulum is going to form a tubular network around each of the myofibril, and it's going to tightly bound them into T-tubules. And like the more expanded chambers of the sarcoplasm rectilium, the SR, are going to be called terminal cisternae. And they're going to lie on either side of the T-tubules that encircle the myofibril, forming a triad. And then these terminal cisternae contain high concentration of calcium ions. It's all really interesting. They all really work well together. Here's more of a 3D model. We see like this very surface layer of the epimecium. We see like the inner part that's surrounding all of the little bundles, the perimeum, and then the inside of the little bundles, what's surrounding even more little bundles are going to be the endomencium. All right, next. So when we talk about the myofilaments, we're going to talk about the different proteins that are in there. So if we go over here, we have thin filaments and thick filaments. And inside of thin filaments are going to be actine molecules. And inside of thick filaments are going to be myosin molecules, which are very interesting. That's the major protein in each of these. And these filaments are going to have something really important to do with contraction, and we're going to talk about that later. What's next? Was I done with this one? No. Okay, wait, no, the structure. So we see right here that the thin filaments have this more Z-line structure, and we see that it comes from this more like stretched out sarcomere. This cannot happen in an intact muscle fiber. When we get to the M lines, we see that these is what is thick filaments is made up of, like the structure is going to be in these M lines, while the Z lines are going to be in thin filaments. Yes. Oh, this is the same image. Yeah. So so the filaments are going to contain the proteins that bind during contraction, and we're going to talk about that in a second. Yes. So this is a relaxed sarcomere showing like the locations of the A band, which are at the top right here, the Z lines, and the I band. This is a relaxed myofibril. And the contracted myofibril are going to be more, the A band was going to stay the same width, but the Z lines are going to move closer together and the I band gets smaller. So, if it's relaxed, it's more stretched out. If it's contracted, it's more like squeezed in. All right, and so this is something called the neuromuscular junction, and it's very important. We're going to kind of like talk about the structure and what happens in it. So I'm going to kind of like bring you through this diagram. And so the cytoplasm of the axon terminal contains vesicles filled with molecules or ACH. ACH is a neurotransmitter. It's going to be a chemical released by a neuron to change the permeability or the properties of another cell's plasma membrane. And so, like, the synaptic cleft and the motor end plate contain molecules of the enzyme, ACHE, or breaks down ACH. That's going to go into the stimulus for ACH release is the arrival of the electrical impulse or action potential of the axon terminal. And the axon potential arrives in the NMJ after traveling along the length of the axon. And these are all kind of like the events in order, just like a brief overview. So when the action potential reaches the neuron's axon terminal, permeability changes in the membrane, trigger for the exocytosis of the ACH into the synaptic cleft. Exocytosis occurs as the vesicles fuse with the neuron plasma membrane. ACH molecules diffuse across the synaptic cleft and bind to the ACH receptors of the surface of the motor end plate. ACH binding alters the membrane permeability to sodium ions. Because of this extracellular fluid, containing a high concentration of sodium ions. The sodium ion concentration inside the cell is very low. Sodium ions rush into the cytosol, and the sudden inrush of sodium ions results in the generation of an action potential in the sarcolemma. ACH quickly breaks down in the ACHE quickly breaks down the ACH on the motor end plate and the synaptic cleft and removal closes the ACH receptor membrane channel. The muscle fiber shown above indicates the progression of the channel. Yes, okay, so it was the diagram up here. So the action potential generated at the motor of the end plate, it now seeps across the entire membrane surface. We see that right here. And if you don't recognize this image from earlier, it's just showing a muscle fiber. It's also, written right there. So here we have the axon. We have the motor neuron, which is just the path of the electrical impulse. Right here we have the neuromuscular junction. And a single axon may branch to control more than one skeletal muscle fiber, but each muscle fiber has only one neuromuscular junction. And at this neuromuscular junction, the axon terminal of the neuron lies near the motor end plate of the muscle fiber. So here we see the axon terminal, the kind of end of it. We see our sarcoplasmic reticulum, our motor end plate, our myofibril. Yes, we talked about that all earlier. So yes. Remember, it's the path of the electrical impulses and kind of like the action potential, and it spreads across like the membrane surface. Right. So now we're going to talk about the contraction cycle. And during the contraction cycle, ATP is always being like reused and recycled. We did mention this before in one of our previous presentations. Like I think either like our second or third one, we were talking about the body systems as a whole. So ATP is going to be hydrolyzed. So remember earlier when we talked about thin and thick myofilaments, the filaments? So they have different proteins, and these proteins are going to be binding together during contraction. We're talking about that now. So first is the binding of the myosin to the actin. We talked about that earlier because myosin is for the thick filaments, actin is for the thin filaments. And so the inorganic phosphate is released. Let's give them a power stroke. We see right here, we have the ADP. And so rigor, which is myosin in low energy form, ADP is released, and the actin gets pulled forward to the middle of the sarcomere. New ATP binds to the myosin head. The unbinding of the myosin and actin happen. ATP is hydrolyzed. And right now is the cocking of the myosin head, the myosin in high energy form, and then it restarts. So I don't have the image from that like third or fourth presentation, but it was like the ADP and the ATP are always getting recycled. They're being reused. It's a continuous cycle. And this binding and unbinding of these thin and thick filament proteins is always happening as well. And so last off, we're going to just talk about a few keywords. And so insertion is going to be the point of attachment of a muscle. It's going to be the end that is most movable, while the origin is the point of attachment of a muscle, which does not change position when the muscle contracts. And we're going to talk about that over here because we have three keywords right here, prime mover. And prime mover is a muscle whose contraction is responsible for producing a particular movement. So like the biceps brachii muscle is a prime mover that flexes the elbow. The antagonist is going to be a muscle whose action opposes the movement produced by another muscle. So like antagonists can also be prime movers, and we see this like with the triceps brachii muscle. It's a prime mover that extends the elbow. Therefore, an antagonist of the biceps brachii, and the biceps brachii is an antagonist of the triceps brachii. Agonists and antagonists are functional opposites. So if one produces flexion and the other primary action is extension, because again, they're opposites, they're opposing each other. And then synergists are going to be a muscle that helps a prime mover work efficiently. And so synergists may either provide additional pull near the insertion or stabilize the point of origin. For example, like the deltoid muscle acts to lift up the arm away from the body. This is abduction. Well, a smaller muscle, the supraspinatus muscle, assists the deltoid in starting this movement. Fixators are synergists that stabilize the origin of a prime mover by preventing movement at another joint. So it's very important that we know where insertion and origin is, because, as we said right here, like synergists, fixators are synergists that stabilize the origin of a prime mover. So we're talking about the origins of some of the muscles. We're talking about the insertions of some of the muscles. They all just really work together. Well, of course they work together because it's a presentation about the skeletal system. Yes. But that is everything. Yeah. And this was really fun. I learned a lot. See y'all next week.